for the introduction. Hi, my name is Hao. I'm from Cornell University. I'm a graduate student in Professor Ilana Brittle's lab, and I'm currently a research intern at Genentech Roche. And today, my title of my talk is Host Microbiome Protein-Protein Interactions Capture Disease-Relevant Pathways. We know that host microbiome plays a critical role in host health. As many studies have shown, microbiota are related to a broad range of human diseases, including cancer, IBD, diabetes. It's fascinating to see that the field is moving beyond community-level association studies to identify important causal bacteria, metabolites, and proteins. A handful of commensal proteins have been shown to be beneficial to host health. The outer membrane protein Mu-P1100, which has been previously shown to prevent obesity by restoring gut barrier integrity. Treatment with pasteurized Mu-P1100 increased the expression of genes encoding the tight junction functions. A commensal protein P9 binds to ICAM2, increase thermogenesis, and GLP-1 secretion, which is a therapeutic target for type 2 diabetes. In a database we curated for putative host microbiome PPIs, we found only 10 out of 5,000 detected human microbiome PPI have structural information. In the absence of structural and direct experimental data, sequence identity methods have been used to infer host pathogen PPI network for single pathogens. But this approach has not yet been applied at the community level for the human gut microbiome. To assess the conservation of interface residues across bacteria members of the same UniRef clusters, we downloaded all PDB structures which contain both human protein and bacterial proteins. The x-axis is the average similarity of human interface residues, and the y-axis is the number of bacteria clusters. We found highly similar bacteria-human protein interface residues between these cluster members, which may allow new PPIs to be predicted through sequence homology. To examine host microbiome interactions broadly, we extracted all experimentally verified interspecies PPIs involving human proteins from large-scale interaction database and a set of manually curated publications. PPIs comprising one UniRef 90 clusters containing human protein and one UniRef 50 cluster containing bacterial protein were retained for like downstream analysis. With within each UniRef 50 bacterial clusters, we further filtered the sequence to bacterial members of the cluster within 70% of sequence similarity to the experimentally verified proteins, and they were labeled as putative interactors. To distinguish PPIs may be associated with health versus disease, we compare community-level PPI profiles in a large case control cohort of well-established microbiome association disorders, including IBD, colorectal cancer, obesity, and type 2 diabetes. To identify host microbiome interactions that are associated with disease, processed abundance matrix of putative human interactors were then to train a random forest classifier on the task of separating disease cohort and control healthy patients. This approach helped us to identify putative disease-relevant host microbiome PPIs. Although the set of experimentally verified interactions, which we call HBNet, includes interactions originating from 82 unique bacteria species, we find that homologs detected in patients' microbiome come from a set of 821 species that better represents the phyla typically associated with human gut microbiome. We found that moving from over 5,000 human proteins within the interaction network HBNet to over 2,000 human proteins with bacteria interactors detected in the human gut microbiome, to 600 that are associated with disease, we observed increasing enrichment for proteins with previously associated or previously reported gene disease associations in CRC, diabetes, IBD and some of the autoimmune diseases. These disease-specific enrichments are even more significant when we look at each specific disease cohort. At the cellular level, microbial protein can act as human protein via several well-established ways. We next examined the localization of human protein targets with the enrichment of genes expressed in epithelium, liver, and blood components in the detected and disease-associated human targets. One of the major advantages of our work is through this new interaction network, we vastly improve our ability to annotate host-relevant microbiome functions. 13% of our disease-associated bacterial clusters contain no member with annotated microbial pathways. Using similar homology searching against the bacteria in these bacterial interactors, most genes can now be annotated according to the pathways of their human targets. Obtaining a putative disease-relevant molecular mechanisms, although these methods generate a hypothesis that directly link human-associated microbiota to disease, functional experiments and structural evidence are greatly needed to confirm these specific host-related functions. Consistent with the idea that bacterial proteins alone can act therapeutically, there is direct evidence for at least 2 commensal proteins which induce physiological effect on the host when delivered by oral gavage, which includes purified amyloid 1100 from Akkermansia and MAM protein from the Calibacterium, which improved host health with glucose intolerance and colitis. We suspect that these may extend to a lot more additional commensal proteins. For example, nanoformestat is anticoagulant that bind complement proteins U1R, suppresses coagulation and fibrinolysis, and provide protection against IBD and CRC. These human proteins are also differentially targeted in healthy patients by our transcriptional regulator SPO0A in lactobacilli and Faecalibacterium. Imatinib targets several tyrosine kinases, including LCK, which is involved in T cell development and has a recognized role in inflammation. Bacterial proteins targeting these same kinases are also consistently enriched in healthy controls across both IBD and 3 CRC studies we included in the analysis. In the future study, we believe structural information is critical to better understand host microbiome PPIs. We hypothesize that knowing the interfaces will provide better information on specific role of protein, as many human proteins have multiple interaction interfaces with other proteins, especially for the differences between pathogenic and commensal proteins. Just to give a few takeaways for our study, we leverage the publicly available interspecies PPI data to find clusters of microbiome-derived proteins with high sequence identity to known human protein interactors. And these methods allow us to mechanistic inference and hypothesis generation from disease-relevant metagenomic data sets. I would like to thank all members of Brito Lab, especially my advisor, Yohan Brito, and co-first author, Wang. I also want to thank colleagues at Genentech who helped a lot on my talk today, including my manager, Alison Bird. And in the last slide, I would like to help advertise the recruitment of the lab. Feel free to email Professor Yohan Brito if you're interested to join the lab as a postdoc or a graduate student. I'm happy to tell you more about the lab and in person. Yeah, thank you so much for your time today. And I'm happy to answer any questions.
Thank you so much. Questions from the audience?
All right, on the way there.
Yeah, take time.
Hi, good morning. Great
Good
talk.
morning.
Can you hear me?
Yeah.
Okay, I'm Liliana Brown from the National Institutes of Allergy and Infectious Disease. Really wonderful work. I wonder if you can explain how much of the data that you used came from the gut, and whether any other sources of data from lung or skin, oral, were also involved.
Yeah, so that's a great question. So from the interspecies protein-protein interaction data we curated from publicly available databases, they're mostly pathogens to pathogenic proteins to human proteins, or like yeast proteins to some other species. But for the specific mapping we did was all about using gut microbiome metagenomic data sets, and yeah, most of the human proteins would be potentially being expressed in epithelium or other important organs in the human body. Thanks for the question.
Thank you so much. I don't see any questions on. Oh, there's a question online, also from Curtis Suttenhauer, and he's asking we were curious about the paper when it came out, and I don't remember from there what you thought. Do you have any type of either computational experimental estimate of the error rates for the approach?
Yes, we're still doing large-scale experimental tests to validate and also validate these predictions we have. And we are doing structural modelings of these results we have. And I think that we will publish these results soon. And also for the PPI database we have right now, and also the way we analyze these metagenomic data set, we are publishing a tool that's coming out soon. Thanks so much for the question.
Thank you so much.